one of the slides we looked at in the last class, we were looking at primer sequences to perform PCR. And I noted that if you have the same sequences on two different chromosomes, these ones circled in blue, those two primers at the same time can synthesize two different sizes of PCR product. Right? The size of this piece on the top we calculated was 14 base pairs long. And this piece that would be amplified, this PCR amplicon with the GT3 allele, there are 3 copies of GT here. Right, so that's the GT3 allele, would make an 18 base pair product. And the whole point of doing PCR and genotyping for us, PCR and gel electrophoresis, is to be able to actually distinguish those two differently sized pieces of DNA. That's the point of gel electrophoresis. It separates DNA fragments based on size. And so we talked about homozygosity. If you have the same sizes of PCR products, either 2 small copies or 2 large copies, those are individuals we call homozygous. And then there's the heterozygote, hetero, that has 1 large copy of this chromosome and 1 small copy of this chromosome. So here's where we left off last time. This is actual picture of an agarose gel with DNA bands. We load all of the DNA samples where? That's the first question. Here's a picture of a gel. Where were all of these DNA samples loaded into this gel? Into the wells. What else? Someone was about to say something. Come on. The top, bottom, left, right of this picture. Right, so the wells are all these impressions in the jello, essentially, holes. We're looking down on the top of the gel, so each of these wells is a depression where we can pipette or load a liquid sample of DNA into. So then which direction from there does the DNA migrate? Down. Yeah. And then lastly, where's the negatively charged terminal? So there's negative, minus is either on top or the bottom. There's an electrical potential put across this gel. So is minus at the top or at the bottom of this gel? Minus is at the top because DNA is negatively charged. So it runs towards the negatively charged molecule. DNA is attracted to the positive terminal. So the samples are loaded into the wells at the top of the gel. The DNA runs. Each of these bands is moving in this direction, down. And the negative terminal is at the top of this gel. Very good. So then I add in details about the molecular weight marker here. This lane or well I've labeled M, marker or molecular weight, whichever one you like, has a series of bands that we know the molecular weights of, or the number of base pairs. That's what's written here. 1,500 means 1,500 base pairs long. 1,000 means that band right there is 1,000 nucleotides long. So of these experimental samples, what's the largest piece of DNA that was loaded in wells 1 through 7? Approximately. 800, something like that.
900.
Maybe 9, maybe 750, I don't know. These are the largest bands. And so if we look across here, we match them up and yeah, it's something like 850 maybe base pairs. 800, 900, reasonable estimates. Okay, good. So this is how to read and analyze and interpret an agarose gel. Right, the one piece of information we get is what size are the PCR products. These are multiple millions and billions of copies of DNA that were produced by undergoing polymerase chain reaction, PCR. So how do these differences in size that we see on these sorts of gels arise? So this is, yet again, how do we get differences in size in PCR products? So I'm going to give you two examples of molecular ways at the DNA level where you can have difference in size between two different chromosomes. This one we've already seen once. So this is review. You could imagine that we have two different worm strains. Now I'm going to talk about C. briggsii because that's my current love and passion. Well, don't tell my wife I said that. My current research love and passion that we work on across the street in Science 1 is C. briggsii, Caenorhabditis briggsii. And all the students in my lab work basically with these two strains. You don't have to memorize this. One of them is called AF16, one of them is called HK104. And in this case, these worms have different sized chromosomes between the same primers. So we could write primer sequences. Let's just write really short ones. So let's say there's an ATG, both in the AF16 version of the chromosome and the HK104 version of the chromosome there. And the same is true for these primers on the right side. So maybe it's TAA and this is TAA also. So there are regions of the chromosome in red that are conserved. Same sequence in both chromosomes. And then in between, there's some sort of difference, obviously. The lines I've drawn representing the chromosomes are different lengths. So the question here is, what causes the differences in length? So you might have a microsatellite where the HK104 version of the chromosome has 4 copies of TA, a TA4 allele. And then AF16 might have 2 copies, the TA2 allele. And if you performed PCR with these primers that are drawn here, you would see that you produce 2 different sizes of product. Which strain has the larger PCR product? AF16 or HK104? HK104. Right. Same PCR primer sequences. It's always ATG on the left, TAA on the right, but it's in between the primers that there's a difference in the size of the 2 chromosomes. In this case, it's because one of them has more copies of this microsatellite sequence, a short repeat. TA, 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 TA.
Dr. Ross?
Yep.
How do you know the difference between a microsatellite and a gene that has just been replicated between those 2 primers?
So how do you distinguish any sort of difference in size between one chromosome and another? If it's a simple sequence repeat like this one, that's a microsatellite. So if it's some 1, 2, 3 nucleotide repeat over and over and over again, that's microsatellite. If it's a different sort of sequence, if it's a sequence that looks like it's unique, it's hundreds or thousands of base pairs long, and it never repeats itself, that might be more like a gene or an insertion of DNA from somewhere else in the genome. Which is an excellent question because that's the next way, the other important way that you get chromosome differences in size. So one is one chromosome has an expansion or a contraction of a simple sequence repeat. Or there's this, the indel. An insertion or deletion. We've talked about large-scale chromosome changes already in class. So you can have pieces of chromosome, large pieces, medium-sized pieces, small pieces, cut themselves out of chromosomes and move them around to different chromosomes. So it might be that there's a unique sequence here that's flanked by, let's say that's our sequence. So there will still be a T here in the AF16 version, but somehow that TATGT has been deleted from the AF16 chromosome. So next to that T, what's on the right side of the T then? If TATGT is deleted, the AF16 chromosome reads T and then A, and keeps going. So we still have these conserved sequences, including the primer sites. They're present in both the AF16 and HK104 versions of the chromosomes. Now this difference in size, like maybe was just suggested, is the insertion of a gene. Or is it the deletion of a gene? Quick show of hands. Who thinks this is caused by an insertion of a gene in the HK104 chromosome? Okay. Who thinks it's a deletion of a gene on the AF16 chromosome then? Nobody wants to vote at all. Okay, so why not? So a few people raised their hands then because they realized they had to. Why the hesitation?
You guys have more information on like, well, just like.
That's always a good answer.
How
No.
like the ancestors were and, you know, like.
Exactly.
What came from what.
Perfect. Like we talked about last class, if you only have two things and they're different, how do you know which is the original and which is the change? That's why these are called indwells, because we don't know whether it was an insertion on one chromosome or a deletion from the other chromosome.
If we are to assume that Occam's razor is a reasonable principle to follow, wouldn't it be more reasonable that a deletion occurred than an insertion, because it's easier to just wash it and delete it than it is to like find something and insert it?
So, if we follow Occam's razor, which is the simplest explanation is the most likely, which is generally an assumption that geneticists are willing to go with, I think, is it more likely that a deletion happens than an insertion happens? That's a valid thing for you to say. I'm comfortable with that. I'm not sure if experimental evidence actually shows that that's true, but if you want to argue that, that's perfectly fine. So if you had this question and you were forced to say, is it more likely to deletion or insertion, that would be a reasonable response.
Couldn't we say that one's more probable than the other, depending on the amount of, I guess, work that it requires for the cell to perform a deletion over an insertion? Because when you said the one that takes the least amount of work, the one that's likely to happen.
So that's right, so the same argument. So what has to happen if there's an insertion versus a deletion? So deletion, presumably, let's see, if this was a deletion, so there's a chromosome here, you have to cut the DNA there, that's step one. You have to cut the DNA chromosome somewhere else, that's step two. Okay. What has to happen, how many steps are there if there's, so then that part gets removed. What if it's an insertion? How many steps does an insertion of DNA sequence take? Well, first, that sequence you're inserting has to come from somewhere. So that requires probably some chromosome breakage to generate a little fragment. Then that fragment has to insert itself somewhere. So maybe we can argue that an insertion takes an additional molecular step. First, you have to generate a DNA fragment to put into a different chromosome, and then you actually have to break that chromosome to insert your fragment. So that might look like this. You've got a chromosome, you break it, you've got another chromosome, which I'll draw with a double line, not to represent anything about the molecular structure. You have to break, and then to insert that in there, there has to be more chromosome breakage there to stick that fragment in. So maybe, for the purposes of this class, I'd be happy with saying that a deletion maybe is easier than an insertion, if you want to argue that. So maybe that's 2 steps. The way I've drawn it here, maybe that's 3 steps. So here's Leo Velasco, biology BS student who works in my lab, sitting at the microscope looking at some worms. He ran this PCR gel. He's interested in studying 2 different types of worms. We cross AF16 and HK104 worms together and make their hybrids. And some of their hybrids are these bigger ones. They're wild type. These are adults, shown here. They're bigger. This is a fertile hermaphrodite. C. briggsii are hermaphrodites, so one individual produces sperm and eggs and makes their own babies, which is a pretty cool thing if you think about it. If you like. But some of their hybrids, so this is a sibling of this wild type worm. So these 2 worms are brother and sister, well, sister and sister essentially. I don't know, do you call a hermaphrodite female? I don't know. They'll call them sisters. They're hermaphrodites. They produce eggs as well as sperm, so who knows? So these siblings are exactly the same age chronologically. They were born at the exact same time. Yet the worm underneath is an adult, and its sibling, which should be also an adult, is still a larval worm. So it's got a disease phenotype. This sibling somehow grows more slowly than its identically aged brothers and sisters, or sisters. So Leo's trying to figure out, what is it about these diseased or D worms? What's the genetic basis of their disease? What mutations does it inherit from its parents and its grandparents that causes it to have this disease? So he ran this genotyping gel, looking at a bunch of different types of organisms. So he took parents, worms from the strain AF16, here on the left, right, loaded them up here in the wells. PCR products are electrophoresing down the gel. He's got some individuals that are HK104, the other parent. So there are two parents, one's AF16, one's HK104. They have differences in their chromosomes like the one we just talked about. There are some regions of AF16 chromosomes where they're bigger in one place than HK104. So we use PCR and gel electrophoresis to tell. In these F2 hybrids, so these are part AF16, part HK104 hybrids. The F2 generation, so grandchildren of AF16 and HK104. He's got PCR products from them as well. So what did he learn from this gel? Which chromosome is bigger in this case? AF16 or HK104? Where do you look to find this information? Left side of the gel, the right side of the gel? Actually, it's both, actually. There's some AF16s over here as well. You can look at either side. So when you look at an individual AF16 worm, its chromosome, the PCR product from its chromosome is that size. HK104 worms have a larger band. Higher up on the gel, it's physically larger. Okay. The same pattern is true over here on the right side where he ran some additional DNA samples from different AF16 and HK104 worms. Okay. So all of the AF16 and HK104 individuals are healthy. So, or wild type, WT. They're normal. So then he took the grandkids, the F2 hybrids, and he's got 3 different individuals. So he took grandkid 1, grandkid 2, grandkid 3. All of these were wild type or normal. And then he took, say, individuals 4, 5, and 6. 3 different individuals that had the disease phenotype. They grow more slowly. So while I take this question, think about what these PCR results might mean about this disease.
Okay, previously you said before that band doesn't necessarily have to mean that chromosome is that size. It could just be multiple chromosomes that are the same size that are just being stacked up and are being pulled by the electrophoresis. So how do you know that chromosome is that size and not just a whole bunch of them stacked up?
Okay, so all PCR products that are a certain size run through the gel at exactly the same rate. So if you see bands that are different in size like this, you do definitely conclude, and maybe there was some miscommunication in the last class, so we should talk more about this to make sure that I didn't say something that I didn't mean to. This you can conclude definitely that HK104's chromosome is larger than AF16's. So differences in position vertically on the gel do let you conclude something about the size of the different chromosomes. So what's different between the wild type grandkids and the delayed grandkids? If you look over here. What's common to all of the F2D diseased worms? They all have only AF16 sized chromosomes. So now we've got a correlation. Every time Leo sees a diseased worm, whose chromosomes does it have? Which grandparent's chromosomes does it have? AF16's. So there's something about being the grandkid of an AF16 HK104 hybrid. If you're a hybrid and if you've only got AF16 chromosomes where this pair of primers is located, then you get this phenotype. This is how geneticists identify the causes of diseases. You start with this correlation. You try to find a region of a chromosome where the genotype, in this case genotype is 2 copies of the AF16 chromosome, correlates, so there's a genotype, it's perfectly correlated with the phenotype. So it's like you could predict, if I told you this worm has 2 AF16 diploid, so if it's got both copies of its chromosome from AF16, you would say, I predict that that worm is going to be diseased. That's what this correlation suggests. This is one way to use PCR and gel electrophoresis to do some genotyping. What types of chromosomes does an individual have? Where you look depends on which primer pairs you use. Fortunately, I don't think we need to go through this. There were a few of you that got this wrong. So kudos to most of you. Which one is the FASTA sequence? A, B, or C? B. Okay. So I felt comfortable asking you because most of you got this right. So the majority wins in this case. Okay. So that was all from the bioinformatics exercise that was due last night. Blast a DNA sequence and tell me something about what you find. Okay. So the bioinformatics part of this chapter then is about BLAST and it's about genome sequencing. So what can we learn from a DNA sequence and then how can we collect new DNA sequences? Okay. So first off, what's BLAST and why do we do it? As I said before, it stands for Basic Local Alignment Search Tool, which means nothing. Please don't memorize that. It has something to do with alignments, though. So what BLAST does is it takes the entire known database full of DNA sequences that scientists from all over the world have put into the database, which it says down the side there, in August was 199 billion base pairs of sequence. It's a huge database full of all sorts of sequences from all sorts of species from all over the world. It takes your sequence that you paste into the search field, and it tries to match that sequence to all 187 million sequences that are currently in the database and find matches. So you're asking, basically, you paste that DNA sequence into BLAST, you're asking, has somebody else before seen this sequence? If so, what species is that sequence in? What gene is this sequence part of? Where in the genome is this gene? So you get all sorts of information out of doing BLAST. And if you want to try again for practice, which I highly suggest, copy and paste this sequence in. See if you can answer all 3 of these questions. What species is it from? Where in the genome is it? What gene is it? So here are some of your results. So some of you have submitted this Google assignment. You sent me a screenshot of the alignment. Wait. So query, here is the alignment. That's the A part of BLAST, Basic Local Alignment Search Tool. This is an alignment showing your DNA sequence, your query, the part you paste in, and how well it aligns with, how well it matches a database entry. So in this case, the database entry is what's called the subject. How well does the query match the subject on this top line? Pretty well, not well. Are the sequences identical? Pretty close to identical. Are there any differences? all of these vertical lines that run in between the query and the subject, when there's a vertical line, that means they're the same. So you just scan along there and you look for gaps. If there's a space, that means there's no match. That at that particular nucleotide, those two sequences aren't the same. So I look across this by eye quickly and I see there's only really one difference. Where's the difference? In the length. So the subject sequence, here I'm circling in red, has a little tiny bit of sequence that's not present in the sequence we pasted in, our query sequence. What is that sequence? Does it have any interesting properties? CTAT, CTAT. Right, this is a duplicate, CTAT, CTAT. So it's 4 nucleotides back to back, CTAT, CTAT, that are in the subject, the sequence that's in the database. 8 more nucleotides than the one we pasted in. So the first thing you should do when you see that maybe there's a duplication of sequence there is look left and right in the sequence. What's true about CTAT? Is it present in more than just the 2 copies? How far does it go? CTAT, CTAT, blah, 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 CTAT. About that far before it breaks down? So, and remember, this sequence on the bottom, the subject sequence, that's a sequence that's in the database. Real DNA sequence that somebody put in the database. What is this subject DNA sequence? That asks the question at the top, what chromosome, what organism, what species? Right up here at the very top. Homo sapiens, that's us, right? Chromosome 5. So on the bottom here, the subject line, that's the sequence of the human reference genome, chromosome 5. What part of chromosome 5? Left side, right side, in the middle of chromosome 5. What information do we have here that helps us figure out where on chromosome 5 we're looking? These digits here give us the chromosomal coordinates. So where on the chromosome 5 sequence, what part of the chromosome 5 sequence we're looking at. So let's see, if I put in some commas here, that's position 150 million nucleotides on chromosome 5. So chromosome numbering is normally you start on the left. If you draw a chromosome from left to right as a horizontal line, the left side is position nucleotide 1, and then you number towards the right. So this is 150 million nucleotides into chromosome 15, reading from left to right, is where our query sequence is from. Human chromosome 5, 150 million base pairs in. So we're looking at a tiny, tiny little snapshot of chromosome 5. And there's a size difference between our query sequence and what's actually in this individual's chromosome 5. What gene, finally? What information, where can we look up here to find anything about a gene? So we've got human chromosome 5, the length of human chromosome 5, total length, that is, 181 million base pairs long. So we're looking at 150 million base pairs in. So we're about 5/6 of the way down the chromosome or so. Here are features. So what is present on human chromosome 5 about at this location? This is the name of a gene, the macrophage colony stimulating factor 1 receptor precursor. Okay. That's the gene that we're looking at, part of the gene. Skip that slide and go to this slide. Okay, this is another one of your submissions. Oh, first, yes, sir, please. I love this question. It's an easy answer. It's not a particularly interesting answer, but thank you for asking a question. This gentleman in the back is modeling something I'd love everybody to do. Anytime you see something, you should all have seen this, if you stared at it long enough, which presumably you were, even if you weren't paying attention. Presumably you were staring at it. You might have seen, hey, wait a second, some of those letters are in lowercase. Why is that? Please ask. What the lowercase means in this case is, because this is a computer and it's really brilliant, or because some programmer has programmed it to be really brilliant, the sequence database has actually gone through and looked for these simple sequence repeats. And so lowercase is how it sort of flags those for us. So anytime you see lowercase in a BLAST output like this, that is the visual clue to us that, hey, the computer has identified that for us as a microsatellite, simple sequence repeat, something like that. So in lowercase here is the full extent in our query sequence of something that the computer identified as something that looks like a repetitive sequence. It's doing part of the job for us. So how does this differ from the result that one of you sent that was on the previous slide? What's different here about that change in chromosome size? How big is it on this slide? There's still a difference. There's still a difference between our query sequence and the subject sequence, but now it's only one repeat. There's just 4 nucleotides present in the database entry that aren't in our query. What does this mean? What sequence is this? What subject? If you look up at the top, where all the useful information is, it no longer says human Homo sapiens chromosome 5 assembly. What does it say now? It's the name of the gene again, the same gene, colony stimulating factor 1 receptor precursor, CSF1 receptor. It's still from human, so we're still looking at the same gene in the same species. The previous slide was chromosome 5, the whole chromosome 5. This is a different subject. It's not the human chromosome 5. It's a version of the SMF gene that's in the database. Why is this different? Those 4 nucleotides, 1 copy of this repeat here. Why is this different than the human chromosome 5 subject?
I think maybe the word says exons, so I'm guessing they removed out the introns, which is the junk DNA, and you only have in the DNA sequence the DNA that's expressed.
So it's true that this says exons, and it's true that those are the parts that are expressed. But in this case, that doesn't explain the difference between the 2 copies of this repeat and 1 copy on this side. So the answer is, human chromosome 5 was on the previous screen. That's the human reference genome sequence. That's the standard human genome sequence. This copy of this gene came from somebody else, not the person whose genome was sequenced. Right? This is evidence that there are genetic differences between you and I, or between person X and person Y. Right? Person X being the person who donated their DNA to have the human genome sequenced, the human chromosome 5 assembly on the previous slide. This individual only has 1 extra copy of this repeat here, not 2. And so one of you just happened to discover this in the database. Right? Screenshot from one of your submissions. There's a size difference on human chromosomes in this region of this gene. Some people have a certain number of copies of the, whatever, repeat that, TCTA. Some of us have a certain number. Others of us have a larger number or smaller number of this repeat in this gene on human chromosome 5. So whose genome is it that we have the sequence of? When we talk about human chromosome 5, the reference sequence, the thing that's publicly available to all the world online as this is the human genome sequence. I asked you this a few classes ago in one of the exit exercises. Whose genome do you think was sequenced? There were a lot of responses from you, which is not surprising. It turns out an anonymous blood donor from Buffalo, New York, where it's cold in the winter and maybe they were bored and were donating blood, provided most of the DNA sequence that we call the human genome sequence, the thing that's in the database as the human genome sequence. What do you think? Fair? Not fair? What do you want to know about this person?
That kind of makes the question then, when you have a reference, like how do you know that is the standard?
Exactly. How do you know that this is the standard? If most of the DNA sequence came from one of the 3 billion of us, is it really the standard? We call it the reference genome sequence. Should it be the reference? What ethnicity was this person? What sex was this person? We can figure out what sex they were because we can find out if there's an X chromosome and or a Y chromosome in the sequence. Right? There's a lot of information we don't know.
Is this person, like, do we know, like, their health issues, or does it have any mutations or things like that?
So certainly this individual has genetic differences between them and every one of us. Every one of us is genetically unique. There are some differences between every single individual human on the planet. No two of us are genetically identical. So how can we use this sequence as a sequence that can help us learn about humans? So the human genome sequence, the one we're talking about now, was published over a decade ago. And it's been a really great resource. In the last decade, most recently, what a lot of scientists have been working on is sequencing additional people. Right? We started with, let's get a genome sequence to start, which took millions of dollars and many years. Now we're in the phase of figuring out how this one reference sequence is different from other people, other cultures, right? Other peoples, different sexes, people with different health issues. We don't know anything about the anonymous donor. Literally. Totally anonymous sample. Since then, there have been a few exceptions where people who are famous, maybe, have paid to have their genome sequenced and they put that information on the internet so we know whose sequence it was. But most of the time, the sequences are anonymous. Why? So I also asked, if you had your genome sequence, would you make it freely available or would you want it to be kept a secret? What do you think? Opinions about whether or not you'd release your genetic information to the world or not?
We'd have information on like potential diseases you carry or whatever.
So is that good or bad?
Well, personally, I would think it'd be a good thing because somebody, like maybe a doctor who would recognize it as a scientist could help you out or whatever, but at the same time, maybe you just don't want that knowledge public.
That's basically what it boils down to. Other comments, thoughts, opinions about this issue? Do you release your DNA information to the world?
I mean, I think that you can, like, sequence your DNA and then have it available for scientific knowledge, like in a database where people that are licensed, like medical professionals, are going to use that for.
Yeah.
Or biologists.
Well, you've seen the database. You're not licensed, but you've accessed it. So
Yeah, I know.
everybody's got your sequence if it's in GenBank. Right. So you're probably an organ donor too, right?
Yeah.
Me too. Yeah. So there's some of us who say, yeah, have the information. It helps society potentially. But then there are others of us that don't want to have our health record potentially in the public domain. Especially, it turns out, one of the big issues is the potential impact on healthcare, insurance specifically. If an insurance company can get your genome sequence and they see that you have predisposition for breast cancer, are they going to cover you? I don't know. I would hope they would, but so that's one of the big issues that always comes up when we start talking about genetic information and privacy. Is there are some negative uses. You could think of maybe some sci-fi movies where if somebody got your DNA sequence, they could like figure out what your fingerprint was and then place your fingerprint at a crime scene and frame you for a crime. That's not possible, but sci-fi movie. You can imagine, right? Okay. So it turns out that after the original DNA sequence, even before the original reference sequence came out, geneticists had already figured out that you and I differ, if we look at 1,000 base pairs of our sequence, you and I differ about 1 nucleotide in every 1,000. So any two of us are about 0.1% genetically different, 99.9% identical. And that's, again, what I've said in the last decade or so scientists have been doing, is sequencing more people and trying to understand. When you look around, of course, there's a lot of diversity in this room. Skin color, hair color, sex, gender, different proficiencies, different body types. How is it that 0.1% of our genomes, 1 in 1,000 nucleotides, cause all these differences that you see? That's the question that geneticists are working on most recently. If we're 99.9% identical, how are we also different? Okay. So that's the spiel about the human genome sequence. It didn't come from, so it came mostly from one person. We don't know who that person is. Probably not representative of humans in general, representative of one blood donor from Buffalo. So these days, how do we sequence DNA? This gets at the last question I asked at the exit activity, I think, last class. What are some issues with DNA sequencing given this fact? So assume up here at the top where it says reference genome sequence, pretend that that's the actual DNA sequence representation of a chromosome. So that's double-stranded DNA, A's, T's, G's, and C's. The way that genomes are sequenced these days is you break that chromosome up into lots of tiny little fragments, like this one I'm circling down here. So that's a representation of one piece of one chromosome. And then what you wind up doing is you only sequence the very ends, the parts that are shown that are the red boxes here. You only sequence the ends, 35 base pairs or so, of each of those pieces of your chromosome. So it's even worse than what I said on the exit exercise from last class. I think I said you get 100 base pair DNA pieces or 1,000 base pair DNA sequences in a row or something like that. These days, for better or for worse, we get tiny little pieces of DNA sequence, 35 base pairs at a time, at random locations across the chromosome. So in actuality, it looks something like this cartoon. You get this huge collection of random pieces of chromosome, and you only get little tiny sequences, the red parts. How the heck do you turn that into the DNA sequence of a chromosome? What tool do we use? We get all these pieces of DNA sequence. Do you think I sit in my office with a piece of paper and figure out how all of those little tiny pieces of sequence fit together to make a human chromosome 5 that's, what, 150 million base pairs long? You feed it all into a computer and you say, computer, you do your job of figuring out how all of those little tiny 35 base pair long pieces of DNA fit together. So in this case, this is what bioinformaticians, people that do, it's kind of like bioinformagic actually, bioinformagicians, if you know how to use a computer. This is what they do. So they write these computer programs that take those individual sequences and they figure out the most optimal algorithms, right, methods for figuring out how all of those little tiny fragments of DNA line up together. This is called a multiple sequence alignment. As you can see, it's multiple sequences, they're aligned. And at the bottom here, which I'm going to circle in red, this program is reporting to us the consensus sequence. So when it looks at all of the different pieces of tiny little fragments of DNA sequence that were generated in the machine, the DNA sequencer machine, it's telling us this consensus sequence is what all of those independent sequences agree is the nucleotide that should be at any one location. Right? So all of these overlapping sequences have 3 T's there in a row. So that's what's reported as the consensus sequence, or the reference sequence, or the actual sequence of this chromosome.
Dr. Al?
Yeah.
That machine just takes the pieces, cuts them up, and then I guess sequence them or sees what the sequence is, and then tries to put them together in a line.
Right. So the human part of it is take a chromosome out of a DNA sample, or a tissue sample, break it into pieces, feed that into the machine that actually detects the sequence of nucleotides on those fragments. And then the computers do most of the rest of the work. So at this point, what was in the rest of the slides for today are a couple of examples. You could try to work through these examples yourself. They might come back on Friday when we do our review session. In the meantime, please help me figure out what to come prepared Friday to tell you about. So the one question for the exit exercise really is, what is it that you feel like, of all the content that's going to be covered on the test on Monday, what is it that you feel least comfortable about? What would you like more practice on? So reflect on what we've done up till this point in a couple sentences, if you will, please. If there's just one thing. Choose the top thing that you're most concerned about for Monday. So hang on just a second, I'll start this exercise. Okay. Now, I've already discerned what one of those things is by talking to a lot of you. So I did prepare a Google assignment for Friday. It's posted now, so you can take a look at that assignment when you want. So that will be due on Friday. There's no reading for Friday. Tonight I'm going to take, for those of you that participated in the exercise about generating a study guide, I'm going to assemble your questions and answers tonight. I will distribute that tomorrow for your use studying for the exam. So your main goal for Friday is to complete that Google Classroom assignment. Bring questions, because with no questions on Friday, I will just be standing here and you will be sitting there, and that'll be it. And I'll send you the study guide key tonight when I put it together.